The Gram stain is a fundamental test performed in all microbiology laboratories. It gives a quick indication of what kind of organism the medical laboratory scientist is dealing with in their culture, and, depending on the results, will dictate what testing will be required to accurately identify the organism. In order to perform a Gram stain, the organism first needs to be applied to a glass microscope slide. A drop of water is placed onto the slide. A colony of bacteria from the primary culture is picked up with an inoculation loop or needle. The tool is then touched to the slide just to the side of the drop of water and stirred into the water on the smear. If there is a visible amount of material on the inoculating needle, there will be plenty of organisms to be seen microscopically once the smear is stained. The smear should be thin enough to be able to read newsprint through. In order to see the individual cells, you must have a thin smear. The slide must be completely dry before the staining process can proceed. To do this, the slide is placed on a slide warmer. The process also fixes the specimen to the glass slide, which is necessary prior to staining. The term fixing the slide refers to the process that adheres the smear to the glass. This allows the material on the smear to be stained to keep it from being rinsed away during the staining process and kills the organisms so they will no longer be infectious. When the slide is dry, it will be warm but not hot to the touch. The glass slide and smear must be returned to room temperature prior to the staining process. If a smear is still warm when it is stained, unusual staining characteristics will occur and the process will have to be repeated. The primary stain is crystal violet, which is purple in color. The first step in the staining process is to flood the slide with crystal violet stain. The stain works very quickly, and most manufacturers' instructions say to keep the stain on the smear for 30 seconds. Then, gently rinse the stain from the slide with tap water. The rinsing should be very gentle, as there is still a possibility to wash the smear completely off the slide. The rinsing should start at the labeled or frosted end of the slide. The next step is to apply the mordant. In this case, the mordant in use is iodine. This is the most important step in the process. The iodine chemically complexes with the crystal violet to make physically larger molecules. The mordant needs to remain on the slide for at least 1 minute. The slide is then gently rinsed with tap water as in the last step. You will notice that the color of the slide will remain purple at this point. The next step is to decolorize the smear. The first 2 steps of the staining process can be done on batches of slides, but when it comes to the next step, each slide must be done individually. If the decolorization agent is only acetone, the reaction occurs very quickly. If only alcohol is used, the step may take several minutes. In our example, this step will be done using a moderate acetone alcohol mixture of 25% acetone and 75% isopropanol. This step is done intuitively, not with exact timing using a clock. The slide is flooded with the decolorizing agent, and as soon as the color stops rinsing away, the slide is immediately rinsed with tap water to stop the process. In gram-negative organisms, the decolorizing step removes the external surface or lipopolysaccharide layer of the organism's cell wall. The peptidoglycan layer is shrunk down until it is thin and brittle and breaks off, and all the color from the stain is removed from the organism. In gram-positive organisms, the thick peptidoglycan layer has compressed, made an armor around the organism, and the purple stain remains. A red counterstain is applied and allowed to remain on the slide for 30 seconds. In our example, safranin is being used for the counterstain. Other red dyes can be used as the counterstain. The counterstain is used to visualize gram-negative organisms that have been stripped of any color during the decolorization step. The counterstain is then rinsed from the slide as in previous steps. It is important to rinse both sides of the slide to eliminate any confusion that may occur upon reading the slide on the microscope. Stray stain particles on the back side of the slide can confuse and interfere with the interpretation of the gram stain results. Gently wipe off the back side of the slide with a paper towel to remove any excess stain that may have been picked up in the process. The smear can be blotted very gently on a paper towel to remove excess moisture, and then it should be stood up to dry completely. You do not want to place it under the microscope, until it is completely dried. If after staining you can't visualize where your specimen is on the slide, you can make a small dot with a permanent pen right where the specimen is on the slide. This dot will provide a spot to focus on at the microscope and help you find your specimen by identifying the field of focus where the organisms can be found. The completely dried gram-stained slide is then analyzed under a microscope using the 10x and oil immersion 100x objectives.